Today's speaker is Dr. Erdal Cosgun, a distinguished expert in biostatistics, statistical genetics, and genomics data science. Dr. Cosgun's career has spanned both academia and industry, from leading biostatistics research at Acibadem University, Turkey, to driving machine learning and genomic analytics at Microsoft Research. With an extensive research experience in computational biology, he brings a unique mix of statistics and AI-driven innovations to genomics. In today's talk, Dr. Cosgun will explore how LLMs, despite their impressive capabilities, require specialized knowledge for complex domains like genomics. He will discuss his team's effort in augmenting LLMs with genomic expertise by leveraging over 190 million variant annotations through RAG and fine-tuning strategies. I think this work not only advances AI application in clinical genetics, but also highlights the broader application of LLMs in specialized biomedical research. Thank you.
Thank you. I'm really happy to present our work in this seminar series, and I would like to thank you to Metin Gürcan and Ibrahim Karabayır for their invite. I hope our conversations and our seminar will be helpful to create a new vision on how to use the LLMs or the AI for genomics analysis. As Dr. Ibrahim Karabayır mentioned, so I'm a data scientist at Microsoft Genomics team in Microsoft Research. So we have been working on the genomics analysis and the genomics domain almost on more than 10 years, and we are trying to create the solutions for our collaborators and the researchers for how to use cloud and how to scale up the cloud resources for the genomics analysis. And before starting to my presentation, so I would like to highlight the project and the paper that I'm going to share is founded by Microsoft Research. It's just for research purposes, so it shouldn't be directly used for clinical or diagnostic use. So please be aware of these kind of disclaimers, because in the responsible AI world, so we need to mention all these details before our presentations. Before starting to discuss and share what we have today, so I would like to highlight why I'm here, why we published this paper, and why we need AI for genomics. The first thing is everybody is using the different AI agents like GPT-4, GPT-4, or maybe in upcoming years GPT-5 and the 6. So these kind of AI agents cannot be used for the genomics, analysis or genomics representation. The reason is they were not trained on the genomics data. And one of the genomics data and the genomics databases that we need for these kind of analysis is the genomics annotation. These annotation databases needs to represent on the AI agents, because they have all the information about diseases, they have all the information about the relation between the specific variants or genes between the diseases. And we have a chance to use the specific AI agents for using these kind of technologies on the genomics, and we use the variant annotation databases on this work because at the end so we can easily use the AI technologies for adding these kind of information to the existing AI agents. There are several feedbacks from our researchers, from the collaborators from all over the world. Hey, we would like to use the genomics annotation information on the AI agents, but we don't know how to integrate these annotation databases. And in this work, we will present and we will show how to integrate your private or maybe public annotation databases to your existing genomics, or sorry, the existing AI agents. And as Dr. Karabayır mentioned, so we integrated 90 million variant information from 5 different databases in this work, and I'm going to show what's the limitations, what's the gain from this work. And I will share this deck with the organizing committee, but we started to share this work as a tech blog in Microsoft websites. And this work is a great example of using the LLMs on genomics, and one of my researchers, Shangjai from Yale Genetics, so she's doing her PhD on Yale Genomics, and we completed this project with her, and we published the first blog in September, late September, and last month we published the paper about how to use the new technologies and the new AI approaches for the genomics annotation work.
We also didn't want to forget about hepatobiliary obstruction, especially given her 20-pound weight loss.
Sorry, could you say that again, please?
I think somebody forgot to turn their microphone on.
Yeah, cool. So as I mentioned, so this is the paper, and you can easily, reach out from the Bioinformatics Advances. This is one of the journals that I really like to follow. And before, starting with the genomics annotation work, so I would like to highlight 2 different approaches that we use on the paper, and probably you will need to use, and maybe you will need to test it out. If you would like to add the genomics information to your existing AI agents, you have 2 options for now. One of them is the fine-tuning of the existing model, and the second one is the retrieval augmented generation, the RAG. And in this work, we tested these two options. And if you don't have any familiarities with the fine-tuning and the RAG, the fine-tuning is an approach that you can prepare your own data with the specific information like genomics annotation or disease information, and you can create a table and just create a label on the columns and say that, hey, this is case, this is control, and these are the profiles from my patient. And you can integrate the table to the pre-trained model on GPT-4 or GPT-4o, and you can use this agent with the information that you are providing. So this is one approach, but at the end, this is just integrating the existing knowledge without investing too much money to your GPUs or investing too much money for the domain that you are working on. This is one of the approach that we tested out. Fine-tuning is great for most of the applications like finance, like the regular the life sciences models, but at the end, another option that we found is a great option is the RAG, retrieval augmented generation. In this applications, the RAG applications, you prepare the similar table that you have on your domain, but at the end, whenever you ask a question to the AI agent, it will use the table that you are providing and retrieve the information from there and use the LLM skill set and create an output for you. So it's a little bit different than the fine-tuning, because fine-tuning is just adding a domain knowledge to the LLMs, but RAG is directly integrate your data set and your information to the outputs. So we tested these approaches on the paper, and based on our reports, so the RAG is definitely okay with the genomics annotation work. Therefore, we recommended to use the RAG instead of fine-tuning until we have a LLM or small language model for using the genomics annotation works, or trained on the genomics annotation work. And in the real world, the medical doctors or the bioinformaticians have a report on the left side. So they have just the specific set of the SNPs and the variants, and okay, it's good. They have the variant names, they have the variant IDs, and the IT teams deploy the APIs and tools for searching these variants in the literature or in the existing annotation databases. Therefore, they need to copy the variant names and paste it and search it in the Clinoir, Nomad, or any other annotation databases. But it's still slow, and you can just search it, and you can search and find results, but you cannot have the reasoning models or reasoning hypothesis creation models. And this is the regular pipeline in most of the hospitals in the US and all over the world. You have the report, you can search it based on the internal tools, or you can search it with the regular web pages. But with our approach, you just ask to your GPT-4 or GPT-4o agent, hey, this is the variant that I found on my the patient report. Could you provide the annotation information? Yes, it provides the annotation information. I'm going to show to you it's 100% accuracy with these results. And you can ask for the further information. Hey, is there any specific variants that I can maybe search for casual variants or any relation with other the diseases? It will provide all the information that you provided in the reg or the fine tuning, and based on our feedbacks on the pilot studies, the medical doctors and the bioinformaticians really like these kind of approaches because they don't need to go to other web pages, search with other stuff, and they can easily understand what they have on their data and how can they connect the existing reports with the existing databases on their own. And before explaining the other details, our focus was the VCF file. The VCF file is the output of the secondary analysis from the genomics pipelines. If you have a raw sequencing file like FASTQs, the CRAM files, you can easily convert them and align it and create the variant tables from the raw sequencing data with the existing pipelines. And we focused on the VCF files on this study. The reason is the variant code format file has all the information about the variants and has all the information about the specific annotation information if you use it. And the reason behind of using the VCF file is this is standard. So this is like a best practices, and you can easily find the VCF files or creating the variant code pipelines easily on your own and have the VCF files. And in this study, we use the VCF files, the gold standard VCF files from ClinVar. It has all the information about the variants, and we added the other annotation information and other annotation databases to our work. But if you are familiar with the VCF files, probably it makes sense for you. So from raw genomics data like FASTAs or FASTQs or the CRAMs to VCF file, then you can use it for the AI agents. In this study, we integrated 5 different annotation databases to our model. The first one was the ClinVar, the second Nomad, the SNPF, SNPSIFT, the GWAS catalog, and the pharmacogenomics database. So all these databases probably has all the information about the variants in the literature or the other studies. So whenever you use the approach that we are proposing today, you will have all the informations without thinking about the hallucination, thinking about the wrong answers, because the reg is probably the best answer for creating your own genomics agent. And we tested out the GPT-4 for the fine-tuning and GPT-4o for the reg. The reason is we are using the Azure cloud for all these analysis because we need cloud power to process the data, to store it in the Azure AI search. And we had an option to fine-tune the GPT-4, and we have an option to use the reg on GPT-4o. And this is the comparison between the 2 different approaches. And if you would like to understand how we did it, it's really easy. So if you know how to create your own VCF file on the genomics world, you just need to annotate your VCF file, then converted it with our pipeline, converted it to a CSV or text or JSON-L, so whatever format you have, but we prefer to use the CSVs. And we prepare these VCF to CSV files and upload it to our cloud storage accounts, because at the end, we need cloud to process the data. In the second round, we need to create an index file to read these information from our AI agent. In the reg, or probably the fine tunings, so you need to create your index file to retrieve the information from the databases that you have. Therefore, we created an index data and index table in Azure AI Search and use the Azure OpenAI. It can be the OpenAI too, by the way, so you can create your own reg information or the reg pipeline on the OpenAI or fine-tuning application on OpenAI, but we use the Azure OpenAI. This is the same OpenAI application. And as you can see in the bottom of the slide, so we just integrated the special format from VCF data and use the indexing options in the Azure AI Search. But if you have any other search indexes or if you have any other search index tools, you can definitely use them. But in our application, we use the Azure AI Search to create an index file from our VCFs. This is really important because at the end, you need to understand how can you provide an information, the retrieval information to your AI agent, and this is one of the ways. You can use search indexes, you can use your own search profiles. In this example, as you can see, we added 190 million variant information from 5 different annotation databases and created a table that we can easily retrieve the data from there and use the LLM skill set for creating the outputs. And if you would like to learn more about the search indexes or creating the index files, I think our paper is a great resource because our approach to convert the genomics data to the AI indexes, I think this is one of the niche example that you can easily replicate on your local clusters or any other cloud environments or the cloud vendors. And in this example, so as you can see, we have all the information about the specific variants, what's the allele frequencies, or what's the reference genomes, what's the search score that can be related with your search or related with your the prompts on the AI agent. This is the data structure that we shared on the paper. We tested out probably more than 10 different data conversion options to use the VCF data, the genomics data, annotation data, and use it as a reg or fine-tuning on the AI agents. And this slide is just a representation of the best choice that you can create your own genomics data for the AI agents. This is the regular in the black side, the black screen. It looks probably very, very complicated, but it's not complicated. We just edit the information to our the VCF file and add the annotation information from 5 different databases and create a CSV file for the Azure AI Search Index. And this is not a classical bioinformatics annotation pipeline. This is one of the unique data format that you can easily create by your own and use the search indexes. And based on our performance benchmarking results, you will have great results and very fast responses if you use the approach that we are providing today. And the results are really interesting, actually, because if you have a chance to test it out, the GPT-4 or GPT-4o, by the way, we have new models, but at the time of the publication, we just have the GPT-4 and GPT-4o. In the left side of the figure, if you search for a specific chromosome and position information, it's real difficult to have a real response and accurate response from the GPT-4 or GPT-4o, because you can ask a gene name, but you will have results with a hallucination, with the wrong results, definitely. But if you ask us well-studied genes, this means very well-known genes, you will have a little bit, better results. But if you ask for a condition, the condition means the diseases and any other information about the genes and variants, you will have a very low prediction and very low the responses, accurate responses. It's same for the DB SNP IDs, because most of the bioinformaticians and the researchers are looking for the DB SNP IDs, like RSIDs, the reference sequencing IDs. And if you ask these kind of the RSIDs or DB SNP IDs to your GPT-4 or GPT-4o, you will have a very low accurate results. So this is the problem. So I cannot use the existed AI agents or existed LLM models. Therefore, I need to add my information to the models. And the first thing that I discussed earlier was fine-tuning. And in the fine-tuning, you need to create a new table and add this table as a new knowledge or domain information to the existed LLMs. And you need to create prompts for these kind of approaches. Let's say in the fine-tuning, we are adding, hey, you're an expert genomics annotator. It's okay. And I would like to have an information about the specific chromosome and specific position in a specific reference genome in the user prompt side. And it will provide a output, and it will provide a response for you. And in this fine-tuning example, the GPT-4o cannot provide the accurate information for the specific variants or specific chromosomes. Therefore, we fine-tune the GPT-4 with the gene information. If you fine-tune it, the model, it will provide a gene name with the correct gene name in a specific position with 0.95. This means 95% accuracy. It's okay. You can fine-tune the model for genes, but genes is just a gene name. It doesn't, make sense for most of the researchers. And we fine-tune the model for the condition information, which includes the disease relation with the variants, genes, or some kind of the literature information about the variants in the literature. And it just, provide the 54% accuracy if we fine-tune the model. And we didn't stop in that point, so we added, hey, if we fine-tune the model with gene and condition information, what should be the, the accuracy? It's still low for the condition, which I really would like to see the real results and accurate results. And we fine-tune the model with several approaches, like gene, fine-tune the model with gene, then condition. It's not, creating the accurate results. We just fine-tune the model with the RSIDs or DB SNP IDs, and it didn't work again. And we tested out a different approach, which is really, important for the bioinformatics world. We create a new encoder to convert the RSIDs to a new model, encoding just using the random words, converting the RSIDs to random words, because LLMs requesting the words and the letters. And we have a great accuracy on the RSIDs. But still, this is just, the slide that I would like to say. Fine-tuning is not the best approach to extract the genomics information from your external knowledge. Okay, we can predict the RSIDs, we can have the information from RSIDs, but it's not going to work because we need more information from the genomics domain or the genomics data set. And after the fine-tuning examples, as probably you're expecting this result, if you use the retrieval augmented generation, the RAG, we have a 100 accuracy because we are indexing the genomics data, the annotation data on the Azure AI search. Then we ask the specific questions about the RSIDs, the disease information, or allele frequencies on a specific position. It will provide the 100% accurate results. So on our example, we integrated the 5 annotation databases, and we are adding more every day. But for now, I don't need the regular GPT-4 or GPT-4o because I have my own RAG file and RAG databases. Then whenever I use the RAG application, I will have the right information that I'm looking for. but this is just maybe the graph for you, but the 100 accuracy is really important for the variant querying or creating the reasoning model from the variants. Please take a look and have your seconds to review this slide. If you are a GPT-4 user or GPT-4o user, you can ask the same question in the left side. They provide the annotation information for variant chromosome 22 on a specific position. It will, the response probably, and it will respond with, hey, this is the variant. You can, find the functional annotation information from specific databases. We say just a general information. So you will not have the accurate information because the models was not trained on the genomics annotation or genomics information. In the left side, after our reg implementation, so it will create the specific report that you can easily use on your daily routine. We edit the nomad, ClinVar, SNP. If your record has or the variant has a record on the reg databases, you can easily have the reports and the right information. And it's not just providing the search results. It's not just providing a report for your analysis. And as you can see in the question, hey, write a brief summary explaining the function of this variant. So you cannot have any response in any AI agent today because they don't know what's the meaning of the function of this variant or some kind of disease relation with this variant. And as you can see in the response, it will use the annotation information that I provided to the agent, like GPT-4o, and at least creating a hypothesis for me. Hey, this is the functional impact because I provided the annotation information and it will create the functional impact summary for me. It's good because if you would like to send an abstract to the conference, or if you would like to create a report, you can easily use the specific responses for the annotation information. And this is the example that we can use. Let's say if you have a specific set of variants, you can easily ask them, hey, I have the list of variants from my patient report, and I would like to have the annotation information, and I would like to report the condition, the symptoms, or specific conditions that I have. And this is really important because most of the genomics reports has several variant list or several candidate variants that they would like to explore. And I asked the same question to my AI agent. Did I use reg? Oh, sorry. Did I use reg? One of the important part of these kind of exploration is you can have the annotation information plus I'm looking for, hey, explain why these variants are important, like the disease casual variants or some casual variants that I'm looking for. And it will provide the information that I put on my reg databases and easily create a reasoning model or hypothesis for the specific variant set that I'm looking for. And as I mentioned earlier, so if you are working on bioinformatics or genomics, definitely these kind of informations are really useful. But at the same time, if you are looking for a new hypothesis or a new idea generation, the LLMs can easily help you to create these kind of the responses. And I have two more examples that you can easily replicate on the diseases that you are working on. The one of them is the sudden infant death project that I'm working with the Seattle Children's Hospital. We have been working on the SIDS, the sudden infant death syndrome, and exploring the genomics background of this syndrome. And we published a paper last year about understanding the genomics variant impact on the SIDS. And I just created the two different prompts for you, because I already published the paper, I already have the candidate variants, but if you would like to, adding these new information from your paper to the AI agents, it's really easy because I have the reg databases. And please take a look for these two different questions. Most of the researchers in the SIDS world, the sudden infant death world, they would like to understand the genomics variants impact on the treatments or any maybe relation with the other diseases. So they have questions like that, and they need to go and search with the the databases, and still they need to understand the relation between the different diseases or different annotation informations in the literature. But with our approach, so this is one of the example. I asked the first prompts, hey, could you provide the details on implicated genes or their biological functions or supporting studies? In seconds, you will have the outputs. Hey, this is the gene that many people, reported on the annotation databases. And if you would like to, find or explore the relation between the other diseases, this is the reference from my annotation rec. And if I don't have, as an agent, if I don't have further information, it will directly say that, hey, the requested information is not available on retrieved data. So this is good because it's not creating a response without any, any dots on the annotation world. It's just creating the results that they have, and the hallucination is almost, zero. So this is good because I can ask the question on a specific disease, and I edit my information to my reg, and it's really important to have the right data and accurate data from my end. And the second prompt is very similar. The polygenic risk scores is really important for most of the genomics analysis. And I focused on the specific disease, and I would like to explore, hey, do we have any insights about the polygenic risk scores in the literature or in the databases that I provided for reg? And it can easily create the methodological considerations or limitations on using the PRS, and it's creating a reasoning model or reasoned responses for me. And in these kind of use cases, the medical doctors or the bioinformaticians, geneticists, just have the information from their private or public annotation databases, and they can easily replicate the same studies for the other diseases. And I just asked the question about sudden infant death syndrome. Just ask the name of the syndrome, and it will provide the list of the genes and provide some kind of reasoning on how to use these kind of information on your study. Or if you would like to continue to the prompts, you can ask for, hey, this is my hypothesis, or I need a hypothesis to work on. This is agent. This is the AI agent that you can easily talk and easily export the relevant information from your use cases. At the end, the conclusion. So as we mentioned earlier, so we tested fine-tuning and reg, but fine-tuning is not going to work, it's clear. Because we have very detailed tables, very detailed information. Therefore, reg is definitely the best choice. And one of the advantages of using the reg is it's really easy to access the up-to-date information, up-to-date research. Let's say you have a genomics project, you created your annotation information or disease-related information, and it's on your databases. You don't need to wait for GPT-5. You don't need to wait for GPT-6. You can easily use your existing table with the reg and have the up-to-date research. And for the variant perspective, there are several interpretation tools in the literature and in the market today, but you can easily create your own information and use these kind of technologies for improving the variant interpretation work. And the cross-disciplinary insight is really important because I'm working with the engineers, I'm working with the bioinformaticians, the geneticists or medical doctors, they always have their own special information. And if I have a chance to merge all these information on the rack, it's really easy to create a new collaboration projects or the new the hypothesis-based projects or proposals, because I don't need to understand all the information on the other domains. I just need to use a RAG or maybe fine-tuning for the other purposes. But for the genomics world, the RAG is the best option to combine all these cross-disciplinary insights. And the fourth and the fifth items are really important for understanding or defining the risk of the patients on having the specific variants or having the specific expression level in the databases. And I don't need to go and search the other databases. And if you are familiar with the AI agent creation, it's really easy. You don't need to use just Azure. You can use any cloud vendor. You can use your own cluster to create these kind of up-to-date and efficient literature mining options for the genomics annotation world. And I presented the similar similar works in the different places and conferences, and these are the questions that I'm always having. And if you already have the questions, I will be happy to answer. But before your questions, one of the top questions that I have is, hey, is the reg option that I'm using is a standard reg, but is there any other reg options? Yes, there are several reg options in the literature. One of the popular one is the graph reg. Many people are looking for the graph reg, or sparse reg is one of the popular ones. Definitely, you can test it out everything. So as the researcher in Microsoft, so we have tons of compute opportunities and compute environments that I can test all of them, but it takes some time. Therefore, if you are looking for the other reg options, there are several of them. So I hope we will have the benchmarkings for all of them in future. And another question that I'm always receiving is the security, privacy, and responsible AI. I'm using Azure, so it's safe for me and for most of the customers. But at the end, if you would like to use the other cloud vendors or local clusters, definitely the security and privacy is really important for genomics data. And I'm working with the security experts and the privacy experts, usually on the collaborations projects or any other internal or external projects. So definitely we need the security and the privacy experts to use the AI on the genomics world. And responsible AI is a great area and the domain. I think if I have a chance to work on the responsible AI work, I think this is one of the opportunities that you can publish more papers, because we need to be transparent on what type of data we used, what type of technology that we are using, and responsible AI principles are really important. And you can easily implement the same technologies, fine-tuning and RAG on your local cluster. It's definitely possible. And the small language models, this is one of the area that I'm working, and I will publish a tech blog soon, because the limitations of using cloud, you have an option to use the small language models like Phi 2, Phi 3 in the Microsoft side, the Phi models. And if you would like to deploy the same RAG or fine-tuning approaches, the small language models, I think, is a great option. And please, take a look and search for the small language models. And there are several genomics foundation models in the literature. But not all of them is really, helpful for everybody. But the genomics foundation models are one of the final points that we need to focus on. And I hope we will have the new genomics foundation models soon in the research world. And for the cost perspective, definitely, if you would like to use the fine-tuning, it's relatively costly. But if you would like to use the RAG, it's relatively cheap. but please, have your, IT people or your cloud responsible person for evaluating the cost, because sometimes it needs more money to maintain the fine-tuning models or maintain the reg approaches. And before I completed my presentation, so I would like to highlight, I'm sure we have some the PhD students in the attendees. So the Microsoft Research is a great research intern project, and you will have the different opportunities on using the AI, using the genomics AI or any other life sciences related AI technologies for these kind of internship projects. So please take a look at the Microsoft Career webpage, and you can easily find the internship program that you maybe would like to apply. And please, feel free to reach out to me for any questions about the research internships or the project that I'm presenting today. So thank you for your invitation, and I will be able to provide more information for you.
Thank you so much, Dr. Coşgun. Really appreciate that. I think it's really great presentation. Latrice, do we have any questions in the chat? If you don't have, I actually have one question I can maybe start with.
Yeah, absolutely. We have no questions in the chat, but after your question, people can feel free to raise their hand.
Yeah. Dr. Coşgun, you experienced the GPT-4 and GPT-4o as a foundational model. I wonder that, have you ever tried to utilize Llama, like, open source foundational model to fine-tune NRAG, for fine-tuning NRAG applications for genomics, for that specific study? Because we know that, lots of people doesn't have access to a foundation GPT-4.
Yeah. So yeah, I don't have specific experience, but I know researchers from the different universities, they are testing out. One of the things that I'm always focusing on is the security and the privacy. So if you have a chance to create an environment for your reg or fine-tuning with the open source models, with a secure, implementations, it's good. I don't have a specific experience, but one of the things that I always recommended is create a secure and private information, because the open source models sometimes needs to reach out the external resources, sometimes it needs to download specific databases for reg. Therefore, I just said I didn't have experience, but I think we need to be sure we have a secure environment for the open source models. I'm not saying they are not safe, but we need to be sure and get help from the IT teams and the security teams.
Yeah. If there is no other questions for now, I can ask one more.
There are questions.
Okay.
They are raising their hands.
I'm sorry.
Yeah,
Mustafa, I see.
Seha raised his hand first, and then Mustafa raised his hand.
Yes, thank you very much for your great presentation. I really like your presentation. So I have two questions for you. The first question is, right now from this presentation, it seems that you are using RAG only for variant annotation. So you're using FASTQ files and BAM files. So I wonder if we can use RAG for like using bulk RNA-seq, like after alignment. This is the first question that I have. The second question that came to my mind during your presentation is using RAG for variant or gene mutation prediction. Have you ever seen any work like this one?
So could you repeat the second question?
So the second question is because I noticed that you are using RAG for variant annotation. I was wondering if we can use it for gene mutation prediction.
Yeah, so let's start with the first question. So using the RNA-seq data or the raw sequencing data is another challenge. So there are several efforts in the literature and the research world, hey, use the raw sequencing data for the AI agents. So my personal opinion, not as a Microsoft employee, so I think that using the raw sequencing data is not going to work because there are several structural variants, several mutations, as you mentioned in the second question. So I don't have any specific example. I don't see any successful implementations, but probably in the upcoming years, so we will see using the raw sequencing data or RNA-seq data to use these kind of technologies. But for now, I think it takes some time to prove the real accuracy of these kind of approaches. For the second question, yes, we can use for mutations. We can use for, if you already have the list of the mutations and the impacts on the diseases, definitely it's going to be a great use case. So I will be happy to discuss further, but definitely it's good.
Thank you very much for your.
Yeah.
Seha, I think you have a question as well.
Yeah, thank you for the great presentation. I have two questions, and I think you answered the first one, but just want to be clear. So instead of uploading my data to the Azure cloud, can I just call the pipeline in my own cloud so I don't have to worry with the data privacy?
Yeah, actually, on the other side, you can deploy your own app with the RAG or fine-tuning and create your own agent, the private agent, and you can use it without uploading the whole databases. Yes, it's possible, but everything depends on the universities and the research centers' privacy rules. So before using and creating the app on cloud and use it local, I think the consultation from the IT teams will be great.
Okay, sounds great. Thank you.
Yeah.
And the second question is, I'm not sure if this is applicable, but let's assume I have a single cell RNA sequencing data and I want to get a cell annotations. Mm-hmm. And what would be the biological rationale behind? Does it look at the marker genes or highly variable genes to provide me the decision in this case?
I tested on the single cells, and if I provided the accurate information to my RAC, I think the responses are really helpful. I can just say for the single cells, definitely it's another area that we should invest more. I hope.
Okay, thank you.
So it's really difficult to cover all the genomics data types on these kind of agents, but for a single cell specific question, I think there's a potential there, definitely.
Okay, thank you very much.
Mm-hmm. You're welcome.
We have one more question in the chat. If you would like to unmute and come on screen, you can answer your question. This is from Ying Chang Sun.
So I don't have any specific information on this hackathon, but I will take a look. So the question is about the hackathon, the AI agents hackathon. So Microsoft is a huge company, guys. So many teams are doing different activities and the different the project. So, but I don't have any insight on this project.
All right. Yassine has raised their hands for the next question.
Hello, Joshkun. Dr. Joshkun, thank you very much for your nice presentation. And it's very useful for genomic research as well. And my main question is the RAG. Like if what happened is we also created another disease model with the RAG, and I uploaded a lot of research papers. And when we check about any treatment drugs, it just gives us a drug that tested on mouse model, but not necessarily the established drugs that used in the hospitals. Then this created a bit headache that we really have to be very careful about introducing, using the research papers. But in your case is you have like very well established databases, so there will not be any mistake. What should we do in this case, if we have research papers that not well defined and if we're going to use a RAG?
Mm-hmm, mm-hmm. Yeah. So importing the papers or importing the specific documents to RAG is very important. Is very controversial because at the end, I use the VCF file. It's a standard file format, so I know what's the content of the data. But in the paper world, or in using the research papers, research reports, so you need to create your own tables for the RAG. So I'm not recommending using the raw documents on the RAG. You need to create your own tables or your own databases based on the papers, based on the reports. So therefore, I'm just talking about the standard data formats that I'm using on the RAG. But for the other paper or the project-based information, I think you should create your own benchmark tables and the standard tables for the RAG. I hope it answers the question.
Yes, thank you so much. Yes, it's a very good presentation. Thank you.
Therefore, many people are asking me, hey, AI is the new thing, so we are losing our job as a data scientist. No. At the end, if you are the expert on the data creation or data preparation, I think you will be the owner of the AI agent. So therefore, if you are an expert on genomics, if you're an expert on economy or whatever domain that you're working on, I think you should create a data resource or the source for the AI agents, and you will be the owner of the AI agent. So this is my personal view.
Following up the Yasin's question, I think, to me, I think fine-tuning would be better approach to kind of use, like a PDF or, not standardized or unstructured data to feed in the model, compared to RAG, because as you just illustrated, RAG is perfectly fine with the structured data, but fine-tuning didn't work. So
yeah, that's right, Dr. Harris. Exactly, that's what we did. Initially, we tried with the RAG and did not work. Then we used the fine-tuning, and then it seems to work better.
yeah, that makes sense.
Yeah.
Perfect. One minor question from my side. I think for the fine-tuning purposes, you only utilize one database, right? Did I miss this point, or you use all the 5 data bases?
All 5 databases, but just focusing on the specific parameters like RSIDs, gene names, and other stuff. Because in fine-tuning, you need to create your own table for adding to the pre-trained model. Therefore, we need to understand, can we have the real accurate response from the fine-tune model for the genes and RSIDs? Because if we succeeded on that, we will have the other information. But we couldn't be successful on even for adding the gene names and RSIDs. Therefore, it doesn't make sense to add the other full annotation information to the fine-tune model, because the cost is one of the important option, because I cannot have or I will not have the real and accurate response for the gene names or RSIDs. So I don't need to add more.
Yeah. I see Oz's hand also.
Hi, thank you, Ibrahim. Thank you so much, Dr. Cosgun. I feel like our timeline is first time aligned. I feel like we have been at the same places at just different times, multiple times in our lives. But it's always knew you, but I think it was today that now we are at the same time at the same place, virtually at least. Like, wonderful talk, thank you. I just want to ask like more like a broad like a question, like what is the, I feel like they are like you are doing like amazing stuff, like amazing research. What are the like industry, academia, what are the collaboration opportunities between like your Microsoft and the academic institutes or like other businesses?
Yeah, I can just talk about the genomics team and Microsoft because we have different collaborations in the industry plus the research, the research centers and the universities. One of the things is, I can give an example, sudden infant death project that I'm running. So we just met with the researchers on the conference, and they said, hey, we have a genomics data and we would like to scale up the analysis and create the machine learning models, create the deep learning models, and now we are adding the AI models. There is no specific way, but the one-to-one contacts and the PI-to-PI contacts is the best option. This is my recommendation. Even if you are working on the Microsoft research team or any other Microsoft positions now, or Amazon or Google, researchers are saying. So PI-to-PI communication is really important, because I can easily escalate the project proposals to my leadership, and they are evaluating the project proposals, and I can have the support, like having the researchers or having the cloud credits, or if leadership approved these kind of collaborations, we have a chance to invite the legal teams for creating the PI proposals. This is important for the research collaborations. Who is going to own the PI? So I think the first step should be PI to PI communication is really important. And then the PIs can easily, in the Microsoft side, can easily leverage the specific opportunities.
Great. Well, thank you. I feel like it may look like we are doing different things, but like very simply, we do a lot of classification model, prediction models. We have true positive, false positives, true negatives. We somehow believe that some of those people, like a bit true positives and false positives, may not be just the model underestimation, model misclassification. It may also be think that actually there is some certain underlying genetic conditions making it predictable for some patients from ECG, for example, but some are not predictable. That's why they both have the same conditions. So what are those genetic component behind the discovery of that would be very interesting, actually.
And the example that you shared is really important, because you can share your machine learning model, you can create your own API, and people can consume your models. But with these kind of RAG approaches, I can embed the machine learning model prediction approaches to my AI agent. And this is something like merging the machine learning models and the outputs with the AI capabilities. I think this is important to maybe explore more. I will be able to discuss. But as you said, using the machine learning or deep learning and adding the AI skill set is going to be the feature for most of the data scientist world.
Well, thank you.
Thank you.
Any other questions for Dr. Cosgun? I think that's it. Oh no, there's one more question from Mustafa. Mustafa?
No, I'm so sorry. I'm so sorry. That was my fault.
Oh, okay. Thanks so much, Dr. Cosgun, for the great presentation. I hope this, can come up with new collaboration between your team and the researchers here in Wake Forest. We are really thankful for your time, and thanks so much again.
Yeah, thanks for the invite. Thanks everyone for joining us today. Bye.
Thank you.